Hi, this is Dr. Pratheesh, Head Department of Biochemistry.
Welcome to Nehru Arts and Science College, Department of Biochemistry, e-learning class. Today we will be discussing about a new topic, genomics and proteomics. First of all, let's see what genomics and proteomics means. Genomics is the study of genomes, which refer to the complete set of genes or genetic material present in a cell or an organism. That is genomics. Now let's see what proteomics is. Proteomics is the branch of molecular biology that studies the set of proteins expressed by the genome of an organism. This is what genomics and proteomics means. Now let's see what is a genome. A genome is a complete set of genetic material, that is DNA, in an organism that include all its genes. As we all know, our genetic material DNA consists of both coding and non-coding regions. Coding region means the genes which code for a particular protein, and there are regions which doesn't code for a particular protein. Both together forms the genetic material, and this genetic material is found in coiled coil formation. There are chromosomes, and all the chromosomes together forms the genome of the organism. Each genome contains all the information needed to build and maintain that organism. To build and maintain means all the activities that are required for development of that particular that is the physical development for that particular organism, and to maintain means all the metabolic activities that are happening within the organism for its survival. So when you take the example of human beings, we all know that we have 23 pairs of chromosomes. It was only after the Human Genome Project. The project started in 1990 and it took about 13 years to complete. So it was completed in 2003. And only after that we were able to understand that our genetic material, that our genome consists of 3 billion base pairs, and in a human cell which has a nucleus, we have this 23 pairs of chromosomes which have information required for all the building and maintaining activities required for the human being to survive. This is a pictorial representation of cell and its components with respect to genomics. And genomics, we are dealing with our genetic material DNA. So you can see that the genetic material DNA is very long. When it got sequenced, we found that it is 3 billion base pairs long. So that is the distance between Moon and Earth 6,000 times. So how will this long genetic material be incorporated within the nucleus of a cell? So what happens is that the genetic material forms coils and coils, and it coils again and again and form compact structures called as chromosomes. And we have 23 pairs of chromosomes, and these chromosomes are packed into the nucleus of our cell. What made genomics possible? The answer is DNA sequencing techniques or DNA sequences, thanks to scientist Fred Sanger, who developed this technique. In order to understand the genomics, we had to first sequence our genetic material, that is DNA. We all know that our genetic material consists of four letter codon, that is A, C, G, T, adenine, guanine, thymine, and cytosine. So by Human Genome Project, we used different sequencing techniques and we found that our total genome size is about 3 billion nucleotides, and this was made possible only with the application of sequencing techniques.
Sequencing was a
What
tedious process in earlier days. That's the reason why, when the Human Genome Project started, it took about 13 years to complete the sequencing process. But now the situation has changed. Now we have a lot of latest next generation sequencing techniques. With these techniques, we are able to sequence a human genome now within 3 to 4 days of time. So this advancement have helped us to gather a lot of information related to genomics.
okay, what does genomic study do? Why do you want to go for genomic analysis? So genomic study mainly helps us to understand the central dogma of life. That is the flow of information from the genetic material to RNA, then to protein. That is through transcription and translation. This process is called central dogma of life. Almost all the biological activities that is happening within a living organism is through this central dogma of life only. Initially, scientists thought that this is a unidirectional process. That is, the flow of information is from DNA to RNA and RNA to protein. But when science advanced and we got more and more information, we found it's not a unidirectional process. There are certain cases where the translated protein have the information required to modify the genetic material. For example, methylation of DNA. That happens due to some protein activity. Understanding this central dogma of life help us to study a lot of biological activities that are happening within an organism. Now let's make a comparison between genome size of different organisms. The genome size and size of the organism doesn't have a relation, because in case of T2 phages, these are viruses, it is about 170,000 base pairs, the whole genome size. In case of bacteria like E. coli, it is 4.6 million base pairs long. In case of fruit fly, it is 130 million base pairs. And Homo sapiens, human beings, the total genome size is about 3.2 billion base pairs. And in case of plants, it is 150 billion base pair. So as you can see, there is a huge size difference in the whole genome sequence of different organisms. So in order to study the genomic aspect of these organisms, we have to completely sequence these organisms. The whole genetic material need to be completely sequenced to study the genomics. This is another chart which gives a clear idea about genome size in different organisms. From this chart, it is very clear that the flowering plants have a very huge genome size when compared to that of mammals, birds, reptiles, etc. Even the amphibians, fishes, and insects have huge size when compared to mammals. Understanding the genome of an organism is very important. The different aspects and different applications related to the genome analysis will be discussed in our future classes.
From today's class, you might have got a basic idea about what genomics and proteomics means. We have discussed a little bit about genomics, and we will be discussing in our future classes about its application and other things. Thank you for.
Genomics is a study of genomes. The word genome refers to all the DNA contained in the cells of our body. It's like a very special instruction manual that is unique to you and contains all the information needed to make you, run you, and repair you. DNA is a code made up of a 4-letter alphabet, A, C, G, and T, called bases. The order of this code changes slightly from person to person to make you who you are. If you wrote down all the letters of DNA code contained in one cell, it would be 3 billion letters long. So if you put together all the DNA in your entire body, you could reach from Earth to the Moon nearly 6,000 times. It coils and coils and continues to coil until it fits into a tightly packed structure called a chromosome. Each of us has 23 pairs of chromosomes, 46 in total in each cell. You get one half of these chromosomes from your mother and the other half from your father. Your body is a very complicated system, which means your instruction manual needs to contain a lot of information. Scientists embarked on the first full reading of this instruction manual in 1990 with the Human Genome Project, which took 13 years and cost $2.7 billion. Since then, we've had huge leaps in technology, which now make these readings much faster and less costly. Scientists want to better understand the unique differences in our genomes. These differences are called variants, and these variations make us individual. For example, giving us different eye color or hair color. Most of these variations have no effect on our health. However, some have been shown to cause or contribute to certain health conditions. Scientists have also discovered that these variations in our genome can influence if we develop a disease, how that disease progresses, and how we respond to medications. Our genome is not the only thing to influence our health. Lifestyle and environmental factors, such as diet, activity levels, and stress, also play a part. The scientists at Genomics Medicine Ireland are studying the genomes and lifestyles of tens of thousands of people in Ireland to better understand how changes in our genome and lifestyle affect our health. We hope to use this information to help prevent and treat disease